Dear students, we are going to discuss an important topic today, which is very much relevant to your syllabus. That is the molecular biology. This is known as the PCR or polymerase chain reaction. So, what is PCR? Polymerase chain reaction or PCR is a technique used to make numerous copies or multiple copies of a specific segment of DNA very quickly and accurately. So, the polymerase chain reaction enables the investigator to obtain the large quantities of DNA that are required for various experiments and procedures in molecular biology, forensic science, then evolutionary biology, and as well as medical diagnostics. PCR was developed in 1983 by Kerry B. Mullis, an American biochemist who got Nobel Prize for chemistry in 1993 for this important invention. Now, before the development of PCR, how this DNA gets amplified, how the scientists or researchers amplifies the DNA, they actually utilizes the bacteria. So bacteria are utilized for the amplification. What they do, they actually introduce the DNA of interest inside the bacteria by using suitable vectors, and when the bacteria multiplies, the DNA of interest also gets multiplied in this way. So as a result, the large numbers of DNA can be obtained. But now it is a totally different thing. In contrast to the earlier methods of amplifying DNA, now a machine is designed which is known as the PCR for to carry out these reactions, and it will give multiple copies of a billion copies of DNA within a very short period of time, within a matter of few hours, and it also gives the DNA sequence. These are much more accurate. Now the PCR technique is based on the natural process a cell uses to replicate a new DNA strand. Only a few biological ingredients are needed for the PCR reactions. So first one is the template DNA strand. Next, a short stretch of DNA of the template strand should be known, that is the flanking region on both sides, on both the strands. Flanking region sequence should be known, because from this known sequence, the primers are designed. So DNA synthesis cannot be initiated by DNA polymerase without primer. So for DNA synthesis, primer is needed. So the primer actually the short stretch of DNA that are synthesized based on the information of the known sequence on the flanking region of the DNA of interest, that is the DNA which we are, or gene which we are going to amplify. So this sequence should be known. Then what we needed the primers. The primers are designed. Then the DNTPs or the free nucleotides, these are needed, and finally the DNA polymerase enzyme, which is very much essential. So the template strand is also known as the coding strand. Here you should know this. Then the DNA synthesis needs, as I mentioned, the template strand, then flanking region sequence should be known to design the primers. The primers are needed. Then the free nucleotides, that is DNTPs, and the DNA polymerase. These four things are essential. And along with this, the variation of temperature is important. So the PCR is a three-step process that is carried out in repeated cycles. First step is denaturation, which occurs 90 to 95 degrees centigrade. Then comes the second step, annealing, where the primers gets binds with the complementary DNA strand, which occurs at 50 to 55 degrees centigrade. And finally the step 3, that is synthesis step, where the DNA polymerase synthesizes the new DNA strand, and it actually adds the new bases over the primer to form the new DNA strand, that is the daughter DNA strand. Now here you can see that this is a diagrammatic representation of the overall process of PCR. So this is the parent strand, these are the primers, this is the DNA polymerase, and these are the free nucleotides, that is DNTPs. Now in the first step occurs the denaturation, which occurs at 90 to 95 degrees centigrade. The parent DNA strands gets melted, and now in the second step, which is called the annealing step, which occurs at 55 degrees centigrade, the primers gets bind with the complement with its complementary sequences, and now the DNA polymerase can extend this primer by adding the new free nucleotides, that is DNTPs, complementary to the template strand. So the sequence should be complementary to the template strand. Now in this way, two new daughter strands are produced. So we get two complete duplex, and this is the final product of the first cycle. So in this way, we can repeat this cycle for 20 to 20 or 40 times to get millions of copies of DNA. So these three steps are again gets repeated. So first is denaturation, then annealing, then synthesis step. Now here you see this is an important application. What is the application of PCR? So the number of copies are actually doubled after each cycle, and for each cycle near about means 5 minutes of hardly requires 5 minutes, and in 25 to 30 cycles we can get sufficient amount of DNA of interest sequence. Now because the DNA form from a wide range of sources are to be amplified, and this technique has been applied in many fields. That is, PCR is used in diagnosis of genetic diseases and to detect the low levels of viral infections in forensic medicines, in criminal investigations, in genetic fingerprinting, then in forensic medicines, suppose we want to amplify the DNA, or want to get or extract the DNA from blood sources or other sources from saliva and even semen. So this PCR can be used as an important tool to amplify the DNA. In paternal disputes also, the PCR can be used. So then it is used in evolutionary studies, that is the 40,000 years old frozen woolly mammoth, its DNA fragments can also be amplified, and this 7,500 years old human found in the pit bog, that is which is commonly known as Iceman, and which is found long back, that is 7,500 years ago. So from these tissues, or means dead remains, even we can amplify the DNA by using this important technique. Now there are certain questions for you, and who invented PCR? I have already discussed the Kerry Monis. Then what do you mean by the Taq polymerase? Taq polymerase is an important enzyme, polymerase enzyme. Taq, T for thermus and AQ for aquaticus. This is Thermus aquaticus is a bacteria which is found in hot spring. As you can see that the DNA synthesis actually occurs at 72 degrees centigrade, so at higher temperature. That's why this polymerase is extracted from those bacteria, so that they can withstand this higher temperature and can perform their activities, that is synthesizing the new DNA strand. So now you see here Next question is approximately how many copies of DNA gets amplified after completion of 20 to 30 cycles of PCR. So it is actually the exponentially. The rate of amplification is exponential, that is to the power n. So initially it is a duplex DNA, then after separation, strand separation, new two daughter strands, that is we get after first cycle two duplex, then in the next cycle you will get 4, then 8, then 16, then 32, then 64. In this way it's get amplified. So I think the concept is clear to you, and what is PCR and what are the basic applications of PCR, who invented PCRs, what are the requirements for PCR, how PCR is carried out. So all these things I have already mentioned, described in detail. So if you had any further questions, you can WhatsApp in the group, as well as you can mail to my email ID. So thank you very much.